We're going to talk about the quantitation of microorganisms and microbial oxygen requirements, which is a very boring title, I know. Real quick though, what you can see in this little image here that's spamming around, this is actually a reaction that involves singlet oxygen, which is a reactive oxygen species. It is kind of the most basic but dangerous form. It is incredibly dangerous. In fact, if you breathed in just a teeny tiny bit of it, you would die. But it is generated naturally in our bodies and just out in the world, like just the oxygen hanging around us. There's some amount that sits in this format. Not a very large amount, pretty small amount. It's not super stable. This reaction is very specific because it just generates a kind of small amount. Anyways, but it's actually really cool. And so this has to do with our whole oxygen requirements lab. There's a little link down here with a video that covers this. There's actually a really interesting video kind of talking about the creation of it and why it's so dangerous. But oxygen just in general forms these things called reactive oxygen species. And this is the reason why we have to have methods of handling us. So this is our cool science thing of the week. It's just a picture, I know. But you should take away from this that this is the singlet oxygen species being generated here. So when we create reactive oxygen species, what ends up happening to our bodies is we can get things like DNA mutations. We can cut DNA. We can destroy proteins. We can alter cell membranes. All sorts of really bad things can accumulate really quickly. And so we have to have methods of handling that if we're going to exist in an oxygen environment. However, if we live in a non-oxygen environment, let's say we are organisms that don't need oxygen, then we don't have to worry about it as much. But some oxygen is required for the basic building blocks of life. You guys remember what DNA stands for, which I know is hard to remember. It's deoxyribonucleic acid. So deoxy means it's lacking one oxygen. That means it's got a whole bunch of other oxygens around it. So you need oxygen at some point in time to make DNA. So if you are a DNA-based organism, you're going to need oxygen somewhere. And so you generate it in small amounts, and so you still have to handle it. So some organisms that are strict anaerobes or mostly anaerobes can sometimes handle very teeny tiny amounts of oxygen species that are reactive, but they handle it very poorly, and they don't handle like atmospheric oxygen like we have on a regular basis. So we're going to talk more about that later, but you should just take away from this nice little scary image in red that this stuff is super duper ultra dangerous and can kill you. Living in oxygen is inherently toxic, so we have to have methods and mechanisms in our cells, both bacteria and eukaryotic, to survive high oxygen levels. Okay. So background stuff. So less about oxygen, more about just the growth of bacteria. So when we want to find out how things are going in a cell or in a system, we want to figure out kind of how many bacteria there are. And there's a couple of different ways we can do this. It's very important for things like analysis of food, water, and soil contamination. So anything that gets sent to the FDA, or any kind of quality control you do for like there's the Skittles or M&M's factory here in Waco. They have somebody that checks that for bacterial contamination, because turns out if you pump a bunch of sugars through a small tube constantly, bacteria like to grow on that tube, so you have to check it constantly to make sure that it's not creating contamination. So that would be a perfect example here. So we have to do this, we have to check this multiple times, multiple different ways, often, to figure out how safe everything is. Same thing with water. Every water system treatment facility in the country, and many countries, has some method that they use to check this. So particularly of importance is the number of live bacteria. We don't really care about dead bacteria so much, although dead bacteria is kind of an indication that there are bacteria there, they're just dead. Live bacteria, it's very, very important for things like antibiotics, disinfectants, antiseptics, the shelf life of products, figuring out how many organisms are generated. A great example for shelf life is for things that are like ultra pasteurized or have very, very low bacterial numbers. So milk is a great example. Milk can have a certain number of bacteria. If it's too high, it'll spoil too quickly. So we keep it at a very low number, and it'll spoil much less slowly until a point where it gets really bad and then rancid, things like that. So quality control, very, very important. Okay, so how do bacteria grow? We put them in a new thing, you just plop them down in some broth, they go, okay, holy cow, everything's terrible. Let's start getting ourselves orientated. Okay, things are okay now, we're gonna start growing. And so we enter a logarithmic growth phase. So it's an exponential set where they're just growing really, really rapidly, and there's really the only rate limiting factor is how many nutrients they have. Now eventually they're going to start hitting an area where they've grown so quickly that they're using up nutrients and they're dying off. They just kind of die from accumulation of mutations or just being older cells. Happens all the time, where they're just killing each other off. So they hit this thing called a stationary phase, where they're dying as quickly as they're replicating. Eventually, given enough time, they will hit a logarithmic death phase, where they will start slowly dying off because they're not going to be able to have enough resources to survive. If you notice, the astute observer would notice that this phase and this phase, this one's much longer. And the reason why is because you can feed off of the dead bacteria. They are, in fact, cannibals, and they will eat each other. All protein is good protein when you're a bacteria. So this phase is going to be a lot slower. That's the reason why, if we that's the reason why that this one's slower, because it takes more time to replicate and get to the exponential phase. But this way, we can kind of survive and hang around just by kind of eating off and recycling the dead parts for longer and longer period. So that's the reason why that death phase goes longer. Okay. In case you were curious. Okay, so how do we count cells? There's a lot of different ways. There's a bunch of them, in fact, and there's more than I cover in here, but these are kind of the simple ones. So this one, which is direct microscopic count method, using a Petrov-Hauser cell chamber. Basically, we take a sample, we stick it in here, and we just pipette the liquid into this cell chamber right here, and it kind of goes into here. And it has this grid pattern, and using this grid pattern, we can calculate how many bacteria are in a certain grid. And by determining how many bacteria are in that grid, we can use a little bit of pre-done math for us, and we can figure out the number of bacteria per mil. Now this is a very, very handy thing, but it's not really used for bacteria as much anymore. We primarily use it for mammalian cell counts because it's much more effective. They're also bigger and easier to see in liquid samples than bacteria, because human cells and mammalian cells are bigger in general. But we can use this for bacteria too. Now the downsides to this, it's a manual counting method, so you have to sit there and pipette it and focus it and then count it and do the math. It takes a bit of time, and it's just laborious. And of course, what's the difference between a live bacteria and a dead bacteria? Well, who knows? Also, it makes it really difficult if the live bacteria likes to move around a lot. As you guys remember from our E. coli, from our very first lab, they move sometimes, and it makes it hard to count things when they move. Easy way to get around this, by the way, is just take a picture with a microscope camera, because then it doesn't matter if they move. Okay. The other method is electronic cell counters. Now, these are actually really handy. They basically suck up cells, and they only allow things of a certain size to pass through this tubing, and then they look for an interruption in current. And every time you interrupt the current, effectively what that is a cell passing through, because the cells do not allow the current to go through, because lipids don't conduct electricity very well, so it just kind of blocks it for a second. So every time you get a little block, it goes beep, and it counts one. Okay? Very easy, very fast. You could be trained to use this in under an hour and be perfectly competent at it. Downside, it costs a ton of money, and it also counts anything that just happens to be cell size. So anything that happens to fit through there and disrupt that counting machine, it gets counted as a cell. So if you have a dead cell, it counts that. If you have a chunk of rock in there, it counts that. If you happen to have a overly viscous liquid that doesn't transmit, electrical current as well, it's going to count that more often. It's just not the best in some situations. So that's a downside for electronic cell counters. There's also these things called indirect chemical methods, and we can do a whole bunch of different things with this. They all kind of amount to the same idea, which is we're trying to count something that's happening with the bacteria and using that to interpret or extrapolate how many bacteria there are. So we can take the dry cell mass. So we can take a sample, we can kill off all the bacteria, dry them out, measure it, and we can say, this is how many bacteria were in that sample. How many live bacteria in the sample? Don't know, we killed them all. That's the downside of that one. We can use the rate of oxygen consumption, which is okay, but it's sometimes really hard to do, especially with organisms that don't consume oxygen, first of all, or consume very little oxygen, and so the rates are much harder to, increases the error rate of that. We can also do the consumption of a rate of specific nutrients. We can measure, how much glucose is being utilized or how much arabinose or whatever chemical, and calculate it off of that. But these are all indirect methods. We're not actually counting bacteria, we're counting something about the bacteria, what the bacteria is doing. So we're not counting bacteria, we're counting something else. Indirect methods always increase error. Okay, so the last kind of slightly indirect method is using a spectrophotometer. This is like one of the most common methods in microbiology laboratories all over the world. Basically, if you don't know what a spectrophotometer does, it's actually really easy, even though it sounds complicated. We shoot light through a sample, usually a blank, and we say, hey, this is how much light of whatever wavelength we shot through is, how much is getting transmitted through. And we put a sample with bacteria, and we shoot the same light through, and bacteria like to absorb at certain wavelengths. And so if we measure how much wavelengths, the drop in absorbance, the drop in transmission, so the absorbance, we can go, oh, this is how many bacteria in that sample because they're absorbing this much light on this wavelength. Do a little calculations, and we figure out how many bacteria are there in the sample. Two big problems with this. It's not a direct estimate. We're just counting how much light's going through, so we're not counting bacteria, we're counting how much light passes through bacteria, so indirect. We're also counting bacteria that have to be in a certain range. They can't be outside this range. So if we're like growing them in a beaker, one of the really annoying things you'll have to do if you're an undergraduate researcher is you'll have to sit there and pipette it into the spectrophotometer and go, can I check it now? No, I can't. Okay, I'm gonna wait a couple hours and check it again. Can I check it now? No, too small. I'll come back tomorrow, check it again tomorrow. It's too high. Great. So you just messed up the entire experiment, which is not good. That kind of thing happens a lot. So it's got a narrow range that it likes to be in. But it is really fast. I mean, 10 seconds worth of work. But you have to get it in the right 10 seconds to do it. Okay, now the last method, which is one of the most popular and most widely used, it's viable plate count method. The reason why is it only counts live bacteria. So that's always a plus. We only care about live bacteria for a lot of things. So this is the big benefit of this. If you guys go on the FDA website or USDA website that's checking water controls and things like that, they talk about these methods and they have specific standards set for them. We count them by calling these things colony forming units, where we say one bacteria forms one colony. So keep that in mind, that's what we assume. Is that 100% true? Probably, maybe, but we call it colony forming unit. So we can't definitively say that, so we call it a colony forming unit to kind of hedge our bets. So it's not one bacteria, but we call it a colony forming unit. So however many colony forming units there are. Downside, it takes about 24 to 48 hours. So you have to grow everything, which is the downside. Also, it's like media intensive, so you have to make lots of plates to do this. So any sample you have has about 4 to 6 plates, and then a whole bunch of tubes you have to use. It takes a lot of resources. It's a little bit of time, but it's the best method for counting live things. So what does it look like? Well, we got our sample, and typically what you'll do is you'll take your sample and you dilute it in something. You might be diluting it into water or saline or broth of some sort. It doesn't matter. So you'll take 1 ml and put it into 9 mL of whatever you're diluting it to. And you're going to take a ml sample of that and stick it on a plate and grow it. But then that's going to be too high. So this concentration, you can't obviously read it. It's just covered the entire plate with bacteria. Can't use it. So we're going to dilute it again and again and again and again and take samples from each of these and figure out the perfect range. Now there is a sweet spot for this. So this number too low, we won't get good results. This number too high, you can see there's big blobs of bacteria. But this one has individual colonies. That's the colony numbers that we're looking for. The range that we want is between 30 and 300 colonies on a plate. If you're below that, you can't use it. We call that too few to count, TFTC. If you're above that, it's called TNTC, too numerous to count. So 30 to 300. So that's actually really fun because you can count throughout things and you'd be like, I don't have to count that because it's too high. That's great. It's always a good time when you have to do less work. Okay. The downside is when you have more than one plate that's in here, you have to average it, which is really obnoxious. Hopefully we won't have to do that, but if we do, you want to make sure to, when you average them, the same power of 10, which I'll explain in a second. So, the worst part about this though, compared to all those other methods, is we have to do a lot of math, and the math isn't done for us, like in the Petrov-Hauser calculations. That one, the math's done for us, we just find it on the chart. This one, we have to do some equations. So you do need to know some equations, how to figure out dilution. So sample divided by sample plus diluent So however much liquid you're putting it into, that's the dilution. Okay, oops. You should also know that 1 mil, 1 gram, and 1 cubic centimeter are the same thing. They're all equal to each other, so keep that in mind. They're all interchangeable, so this is actually really helpful when we're trying to figure out how many bacteria on a gram of hamburger meat. We can just blend it up and put it in some water, and mils and grams are now interchangeable for that purpose. Okay, CFU per mil calculation is number of colonies divided by the amount you plate times dilution. Okay. So let's take our first one. We'll say we're going to take 1 mil sample. We're going to put it in 99 mils of water. I always label everything because it helps me keep track in my head. I forget, so I highly recommend you guys do this as well. So I got 1 mil of my sample. I'm going to put it in 99 mils of water. So 1 mil of sample divided by 1 mil of sample plus 99 mils of water is 1 over 100. Put this into exponents. That's 10 to the negative 2. Okay. So the bigger numbers on bottom, that becomes negative, so 10 to the negative 2. We're going to call that our mix A, and we're going to do it again. We're going to take 1 mil of our mix A, and we're going to add it to 9 mils of water. So 1 mil of mix A divided by 1 mil of mix A plus 9 mils of water is 1 over 10, which is a 10 to the negative 1 dilution. But we care about what that is from the original sample, right? We don't care how dilute this is from mix A. We care about how dilute this is from our original sample. So to do that, we multiply these exponents together. When you multiply negative exponents together, you just add them. So that becomes a 10 to the negative 3 total dilution. So each time you go down, you have to multiply them together to add them all up. Okay, key thing to keep in mind. 1/10 is equal to 10 to the negative 1. It's also equal to 0.1, which will come in handy later. So keep that in mind. Okay. So let's do an actual, this is an experiment that we'll do. We're going to take some sample, we're going to take 1 mil, we're going to put it into 9 mils, that's a 10 to the negative 1 dilution. That's going to be our mix A. And we're going to do that again. And we're going to do that again. So these are each 10 to the negative 1 from each other. This is 10 to the negative from 1 to this is 10 to the negative 1 from this is 10 to the negative 1 from this. We're going to go, okay, but what's the total dilution of each one? So if we took something out of mix A, we know that mix A is 10 to the negative 1 from the sample. If we took one from mix B, that's 10 to the negative 2 from the original sample. And if we took something from mix C, that's 10 to the negative 3 from the original sample. So let's say we take a mil and we plate them on colonies, on plates. First one, too dense, too many bacteria, over that 300 number, can't count it. Get rid of it. The next one is 158 colonies. That's within that 3,300 range. Yes, we can use it. The next one is 38 colonies. That is also within the range. I know that's not 38 colonies, but I didn't want to draw a whole bunch of dots, so just chill out, okay? And there you go. So now we know how many colonies are in there. We can do the math for viable plate count. So CFU per mil. 158 colonies divided by the amount we plate times our dilution. Remember, we got this plate. It was from the 10 to the negative 2 dilution from the original sample. So 1 times 10 to the negative 2 is just 10 to the negative 2. That's 158 over 10 to the negative 2. When you divide by negative exponents, you just put them up on top as positive. When you put these together, you want to do this in scientific notation, so you move the decimal place over. You are allowed to make that noise in class, by the way. That's the sound decimal point to make. So you move it over a little bit. And when you do that, you have to add numbers onto the 10 to the power. So that's 2, so 2 plus 2 is 4, so it's 1.58 times 10 to the 4th units CFU per mil. Units are very important. And whenever you guys do this math, especially when you're turning something in, it's always a good idea to put a circle around the answer that you want us to look at. This is your way of saying, hey, yes, I did all this math. This is the answer. Please look at this. Don't look at all the other scratch stuff where I screwed up. Because we all screw up. I'm terrible at math. I didn't become a biologist to do math, and yet I end up having to do a lot of math. So keep that in mind. Make sure you put a little circle around what you want us to grade. That's the best way. A square is probably better. Okay, so we go back to this. We go, okay, well, we still got another plate that's usable. So let's look at our 38 plate. We have 38 colonies. We have 1 mil plated. We know we took this from the mix C, which is 10 to the negative 3 from our original sample. 1 times 10 to the negative 3 is 10 to the negative 3. Divide by negative exponents, you put it up on top as positive. We want scientific notation, so we move the decimal place over 1, and that becomes 3.8 times 10 to the 4th CFU per mil. Put a box around the answer. Okay, but we've got 2 usable plates, so we have to average them. Whenever you average them, you have to make sure the power of 10 is the same. If it's not, you can, like, let's say this was a 5 or something, we could move the decimal place over another 1, or move the decimal place over here, so it'd be 38, and that would be 10 to the 4. You don't have to worry about that. You probably won't come up that issue in class, hopefully. And what you guys do to average them is you just add these numbers up and divide by 2. So 3.8 plus 1.58 is 5.38, and then you divide by 2. Terrible math stuff, and you come up with 2.69 times 10 to the 4th CFU per mil. Oops, and put a square around it, which I didn't do because I'm bad at PowerPoints. Okay. Now I know that's really complicated and obnoxious, right? Takes a little bit of getting used to this. So please, if you guys aren't sure about how to do this, speak with your TA, speak with me, shoot me an email. If you're still confused, this is something that you will have to do on the lab practical. So make sure you guys know how to do this. You also need to know how to do this for the pre-lab quiz. So make sure you guys know how to do this. Okay, what you guys will be doing in actual class. You guys will take a colony from the plates that we made this week. You're gonna jab a straw in here. And the way you do this is you jab the straw in, you put your finger over the top, it creates suction, you pull it out, put it in the microtube, and you just kind of squeeze out a little bit of air to make it go up there. Don't put your mouth on the straws, no matter how much you want to. They do not taste good, and that's a violation of safety protocols. So you put it in there, it's already got some water in there that you guys placed, and you're going to vortex it. And you are going to vortex the snot out of this thing. So you guys will put it on the vortexer and it'll spin. Make sure, of course, to put the cap on, otherwise it shoots all the water out. And it's just going to break everything apart and get the colonies out in the liquid. Once you do that, for like a minute or two, once you do that, you will then take 1.1 mils, which, if you guys remember, 0.1 is the same thing as 1 over 10, or 10 to the negative 1. You're going to take 0.1 mils, so 100 microliters, and transfer it over to a tube that already has 900 microliters in it. So you guys will fill up all of these tubes with 900 microliters first. You only need to use one because they're sterile. You can just use one tip for all of your 8 tubes, or 9 tubes. So fill up all of your 9 tubes with the sterile saline first. Then you'll transfer over 0.1 microliters into one tube. Mix this around for a little bit, let it get evenly distributed, and then do it again. Same thing, do it again and again and again, until you get all the way to 10 to the negative 8. So you're going to be using 10 to the negative 8 dilutions. Then you're going to take some plates, and you're going to take 100 microliters, put it on a plate, and you guys are going to smear it about with this guy, which is a cell spreader, or hockey stick, depending on how you like to phrase it, and you're just going to smear it around. Don't press too hard. This is almost like the loop, just like kind of the weight of the plastic, and just gently spread it around so it gets an even distribution. When you do this, it gives us the best results if you have a light touch. So you're just spreading the liquid around so it's nice and evenly distributed, and we should get some colonies. And so then we'll do our CFU per mil thing, which is number of colonies divided by your total dilution. So remember how we took 0.1 mil out of this guy, right? This is a 10 to the negative 5th dilution, but we took 0.1 mil, and if you guys remember, previously we talked about it, it's mils plated times dilution, so 10 to the negative 5 times 10 to the negative 1 is 10 to the negative 6. So this becomes a total 10 to the negative 6 dilution. Key thing, make sure you guys remember. Okay, oxygen requirements. So there's lots of things that need to breathe, everything, except for when it doesn't. So these are some examples of things that don't. This is Clostridium sporanges. It forms spores. It doesn't need to breathe as a spore. It doesn't really need oxygen all that much anyways. This is, of course, a water bear or a tardigrade, depending on what you think. They can survive out in space without oxygen for like really long times. They're pretty cool. And of course, zombies don't need air, and they're underwater, so keep that in mind. Okay, so these are the general classifications of oxygen requirements. You're an aerobe. You need oxygen or else you're dead. So we are aerobes, we need oxygen or we die. Strict anaerobes, no oxygen or else you die. So if you get exposed to oxygen, you're just going to die. You can't survive, you just fall over dead. Facultative anaerobes, they don't need oxygen, but they aren't harmed by it. They can use it. So they have the strength of these two, but none of the weaknesses. So they're like Blade. If you guys have seen the movie Blade, vampire with the strength. No? Okay. That's an older movie. I realize that you guys probably might not have seen that, but it's a good movie. You guys should check it out. Okay. Microaerophiles. Now, these guys don't like oxygen, but they can tolerate some, and they need a little to do their job. So they need oxygen, but they can't have a lot of it. So just, the Goldilocks zone. They just need a little bit of oxygen to do their job, and they're fine. But they get too much, then they die off. The other option is aerotolerant. They can handle some oxygen, but they don't need it to survive. They don't use it for anything. They can just kind of deal with it. And so aerotolerance kind of just, okay, they just kind of hang out and they're like, fine, if there's oxygen, it's not gonna kill me, but I don't need a lot of it. I really don't want it, but okay, there you go. So the big one is this guy, facultative anaerobes. To keep in mind, they don't need the oxygen, so they can survive without it. But if it's there, they can in fact use it to make energy and do the things they need to live. So they're kind of the trick here. They've got the benefits of both of these. Okay. So we're going to do these things called, there's a couple different techniques you can use to grow these things in. One of them is this stuff called BHI augers, which is what we'll be using. There's also fluid thioglycolate. And what it does is it allows bacteria to grow in the zone that they prefer the most. So if you are aerobic, you will pretty much only grow on the top because you need access to oxygen, so you'll only grow on the surface or the top layer. If you're a microaerophile, you will probably only grow in the middle here, just a little bit. So just kind of close to the oxygen, but not much. Last one is strict anaerobe. You will only kind of grow at the bottom. And even though it's this whole green shaded area, it's actually mostly just like at the very bottom. Like they want to get as far away from oxygen as possible. Last one is facultative anaerobes. They will grow through the entire thing, but they will be very heavily concentrated at the top because they like that oxygen. Oxygen is really good at generating a ton of energy. And so they want to use a lot of energy, so they'll have the best growth at the top. If you fermentation requires a lot more energy. It's a little bit slower going. So there'll be less growth throughout here, but you'll still see that growth. And remember, there was that last one that we talked about, which is aerotolerant. Aerotolerant will also grow throughout here, but it'll be kind of a weak growth throughout the entire tube. It won't be very strong. It'll be kind of this light growth. So you guys might see that, you might not. We'll see when you guys actually do these. Okay. What the procedure is, you guys are going to take some tubes out of water baths, one at a time, because they have to stay molten in order to do this experiment. So don't take all of them out, because you won't be fast enough. So take one out that's molten. We'll give you guys a rack to carry them in, because they're going to be larger than average tubes. You're going to add 200 microliters of your stock culture, whatever it is, into here. And then you guys are going to spin it about. Not like this. That's bad. Like this. So you'll rub it back and forth in your hand so that it evenly kind of distributes throughout the entire thing. Now it is it's set to 50 degrees Celsius, which isn't insanely hot, but it's a little hot. So be careful not to burn yourselves or drop the tubes while you guys are doing this. It helps if you rest it on the glass on the countertop and then rub it back and forth like that so that you can kind of toss it between both hands without spilling it. Also, if you do it up in the air like this, the likelihood of you going like this and having it drop and hit the ground is really high, so let's not do that. Okay, let's just do it in our hands on the tabletop so it doesn't spill everywhere. Okay, so once you do that, then you'll let it cool. Once it's cool and chilled, you guys can put it in 37 degrees Celsius incubators, let them grow, and then you guys will check them and see some results. And the results will look something like these. So you guys can see strict anaerobe will only grow at the bottom. These ones are both facultative. I know you can't see the tops of them because the person who took this picture was an idiot. It actually wasn't me. I just stole this from somebody else, so I can't really complain too much. But it's growing throughout the entire tube here and growing throughout the entire tube here. This guy is actually, I think this one is an aerotolerant one, and so you can see that it's growing through most of the tube, but not kind of at the top. So it grows out the entire thing, and this one's a special one because it's a type of coci, and the coci like to form little pockets. So you guys might actually see that in lab. You might not, because sometimes it doesn't like to do it. It just depends. So you guys might see these little pockets of little chunks of bacteria growing in there, or not. It just depends. Okay. So, let's see. Good on time. All right. A couple things. One, make sure you guys are knocking out your pre-labs quizzes ahead of time. They will be due on Monday at 8 a.m., so unlike this week, which was all weird. And again, I apologize for the people that kind of like took it early and then like had a 0 questions. I don't know why it did that. As far as I can tell on Canvas, it's now working. So everybody should be good this week, but make sure you take them. Make sure you guys read through so that we're all on the same page and we can all get done efficiently in lab. Does anybody have any questions? No. If you do, come by and see me after lab, after class. You guys have a wonderful weekend, and I will see you all later.